Welcome, everyone. Have you ever really stopped to think about what goes on behind the scenes with the medicines we take? Like the real calculations behind the risks and the benefits. It's actually pretty complex stuff. Today, we're going to dive into this fascinating report from the ISPure Risk Benefit Management Working Group. It's called A Review of Quantitative Risk Benefit Methodologies for Assessing Drug Safety and Efficacy. Yeah, our goal here is to sort of pull back the curtain a bit, see how the industry and regulators like the FDA and Europe's CHMP are trying to make these decisions less subjective, more objective, more transparent. So the report kicks off by pointing out a pretty big challenge, right? Historically, this whole quantitative risk benefit assessment thing, RDA, hasn't always been done consistently or, presented in a way that's easy to grasp during drug approval.
Exactly. And this isn't just theory. We have to remember the context. In the last, decade or so, we saw major drugs pulled from the market. Rofecoxib, troglitazone. Those situations really hammered home that we needed better ways to assess risk versus benefit before things go wrong. So regulators, FDA, CHMP, they've been actively pushing for more systematic approaches, even getting creative with it.
Okay, creative approaches. What does that actually mean? The report looks at, wow, 12 different methods. We obviously can't cover all 12 right now, but maybe we can touch on a few key ones. Like, what are the main types? Let's start with maybe the ones that sound simplest. Number needed to treat, NNT, and number needed to harm, NNH.
Right. Those are definitely fundamental.
Yeah.
So NNT, it's pretty much what it sounds like.
Hmm.
How many patients do you need to treat with the drug for one extra person to get the benefit compared to, say, a placebo or another treatment? And NNH is the flip side. How many patients need to get the drug for one extra person to experience a specific side effect, an adverse event? They're widely used, yeah. They give you a quick snapshot. And, generally, if your NNT is lower than your NNH, that's looking pretty good.
Right. That makes intuitive sense. Lower number to get good stuff, higher number before bad stuff happens. But here's where it gets tricky, I guess. Those numbers, NNT and NNH, they don't really capture how much a patient values that benefit or how much they dislike that harm, right? Like, is avoiding a headache worth a small risk of something serious?
Precisely. That's the limitation. And that's where some of these other more sophisticated methods come in, like Q-twists.
Q-twist.
Yeah, quality adjusted time without symptoms and toxicity. And there's also INHB, incremental net health benefit. These methods use something called QALYs, quality adjusted life years.
QLLYs, right. I've heard of those. It's like trying to measure quality of life, not just quantity.
Exactly. A year in perfect health is one QELY. A year with, say, debilitating side effects might only count as half a QELY or less. So Q-twist and INHB, they try to weigh the time gained from treatment against the time lost to toxicity. But they adjust it all for quality of life. They've been used quite a bit in cancer research, for example, to really compare treatments beyond just survival rates. Like how much good quality time does this drug actually buy?
Okay, that feels much closer to capturing the patient experience, especially for long-term conditions. But what about just asking patients directly? What trade-offs are they willing to make?
Well, that's another whole category. That's where you get the stated preference method, SPM. Sometimes it's called MA, or maximum acceptable risk. This involves doing surveys, basically asking patients, okay, here's the potential benefit. How much risk of this specific side effect would you be willing to accept to get that benefit?
So you're directly measuring their tolerance for risk.
Yeah, you're trying to quantify those really difficult personal trade-offs directly from the people who'd be making them. It brings consumer choice right into the assessment.
Interesting. So we have these different tools, simple counts like NNT, NNH, value-adjusted time like Q-twist, and direct patient preferences like SPM.
Right. And what's really fascinating, and maybe the key takeaway from the report, is that there's no single best method, no gold standard yet. Each one has strengths, weaknesses, situations where it works better.
So you can't just pick one and run with it.
Not really, no. The report strongly suggests the real power comes from using multiple methods, looking at the drug from different angles, using different RBA tools. That way you sort of bound the risk-benefit profile, you get a more complete picture, reduce the chance of subjective bias creeping in, and help regulators make decisions that are hopefully more objective and transparent.
Okay, so putting it all together, what this deep dive really shows us is this ongoing effort, right? Scientists, regulators, they're pushing hard to make these crucial decisions about drug safety and effectiveness much more rigorous, moving away from just gut feelings or, incomplete pictures towards evaluations that are driven by data, that are transparent, and importantly, that try to incorporate what actually matters to patients. And maybe that leaves us with a final thought to chew on. As these methods get better, and as your preferences as patients get woven more directly into these analyses, how might your own personal values, your own tolerance for risk, start to reshape medicine itself and the kinds of treatments that become available down the road? Something to think about.